Welcome to this explainer. Today we're diving into an invisible war that's been raging all around us for literally millions of years, and we're going to explore how humanity basically stumbled right into the crossfire. You see, antibiotics, they aren't some modern human invention. Not at all. Microbes have actually been producing these chemical weapons for eons just to gain an ecological edge over each other. We humans just happened to discover this wild world of microbial biochemical warfare back in the 1940s.
And that brings up a massive, absolutely crucial question that's on the minds of global researchers today. Are we entering a post-antibiotic era? Because microbes are incredible adapters, the drugs we rely on every single day are facing some seriously terrifying challenges. But to truly grasp the modern threat of these highly resistant bacteria, we first have to thoroughly understand the nature of the weapons we've been using, where they came from, and how they actually work.
Fundamentally, antibiotics are natural poisons. They are designed by nature to kill. Fortunately for us, bacteria have very different biological structures than we do, which allows us to poison them without harming ourselves too much. Now, when a patient is sick, doctors face a really critical choice. They can either drop a broad-spectrum bomb to wipe out a wide range of unknown organisms, which is something they often do early on, or once lab tests actually identify the culprit, they can wait and deploy a narrow-spectrum sniper rifle. And that sniper rifle is highly preferred. Why? Because a broad-spectrum blast will also wipe out all the beneficial microbes that naturally colonize and protect our bodies.
Over just a few decades in the mid-20th century, we rapidly borrowed these ancient weapons. We went from just identifying these natural poisons to harnessing them for global medical use. This era, particularly the 1950s and 60s, became the golden age of antibiotic discovery, launching the two most prevalent classes of antibiotics we still rely on today, the penicillins and the cephalosporins.
Now, science is driven by fascinating people, right? And it actually took a highly unlikely triad working under super austere conditions in a cramped Oxford lab to make this happen. First, you had Ernst Chain. He was a prodigy with an awe-inspiring memory, but he was actually torn between a career in science and being a concert pianist. Then you had Howard Florey, this astute administrator who could mingle with influential people to secure vital funding. And finally, Norman Heatley, a modest, mild-mannered techno-nerd who just jury-rigged all the extraction equipment. Together, they pulled off one of the most outstanding milestones in human history. They were the absolute first to isolate, purify, and utilize a living organism's defensive weapon, penicillin, to treat a human infection.
But they faced a massive hurdle. The mold they were using in England yielded incredibly minute quantities of the drug. With World War II raging, operations were moved to a much safer, well-funded location in Peoria, Illinois. And the true breakthrough? It came from an unheralded bacteriologist named Mary Hunt. Tasked with scouring local markets for moldy produce, she stumbled upon a rotten cantaloupe hosting a wildly vigorous strain of mold. That fuzzy growth, Penicillium chrysiogenum, ended up yielding massive amounts of the drug and became the literal ancestor of all modern penicillins.
Meanwhile, over in war-torn Sardinia in 1943, another major breakthrough was happening. Dr. Giuseppe Abruzzo, a brilliant but very private public health director, noticed something highly unusual. People swimming in the sea near a raw sewage pipe weren't contracting typhoid fever. No way, right? Well, he deduced that something in the water was actively killing the typhoid bacilli. Through his experiments, he discovered an entirely new fungus producing a completely different class of microbial weapons, the cephalosporins.
Part 1. Sabotaging the bacterial wall. So how do these incredible, stolen weapons actually kill the enemy? To understand that, we have to look at the enemy's armor. Bacteria are tiny, but they are packed with material, which results in extremely high internal pressure. To keep from bursting, they build a highly rigid cell wall out of peptidoglycan. It works exactly like a chain link fence. The NAG and NAM carbohydrates act as the long circular poles of the fence, but the real strength comes from the connecting fencing in between. Specifically, there's an unusual right-handed amino acid called D-alanine, and that is the vital link holding the entire structure together.
And that brings us to the crucial point of failure for bacteria, the beta-lactam ring. This highly unstable 4-carbon ring is the destructive heart of both penicillins and cephalosporins. Its exact shape is perfectly primed to pull off a lethal case of mistaken identity.
It acts as the ultimate biological Trojan horse. The beta-lactam ring closely mimics the shape of that vital D-alanine we just talked about. So as the bacterial construction enzymes are frantically stitching the cell wall together, they mistakenly grab the drug instead of their own structural amino acid. It's exactly like a builder picking up bricks made of straw instead of clay.
The enzyme gets completely gummed up, the vital cross-linkages fail, the foundation crumbles, and the high internal pressure causes the bacteria to violently rupture and die.
But wait, there is a secondary, massive killing spree caused by these drugs. Bacteria possess a chemical communication system called quorum sensing. When their populations get too dense and threaten to consume all the available nutrients, they can actually initiate autolysis, a programmed mass self-destruct sequence to ensure at least some of them survive. Well, it turns out, the broken fragments of cell walls shattered by our beta-lactam drugs actually trigger this autolytic system. This causes massive swaths of the bacterial population to essentially commit cellular suicide.
Part 2. The bacteria strike back. Now, it sounds like our drugs are an unbeatable strategy, right? But remember, microbes have been fighting this biochemical war for millions of years, and they were fully ready for our attacks.
Very shortly after penicillin entered widespread use in the late 1940s, doctors started noticing strains of Staphylococcus aureus just surviving the treatments. Why? Because the bacteria were deploying a defensive enzyme of their own called beta-lactamase. Since our drug's 4-carbon ring is highly unstable, the bacterial enzyme simply introduces a hydrogen ion from water right into the ring. This process, called hydrolysis, completely shatters the heart of our antibiotic before it can even do its job.
So humans fought back in the laboratory. Chemists discovered that by heavily tweaking the chemical side chains attached to the drug, they could alter its overall three-dimensional shape. By adding a bulky methyl-containing benzene ring, they created a physical shield. This blocked the bacteria's destructive enzyme from ever reaching that fragile beta-lactam core. This incredible countermove resulted in a brand new weapon called methicillin, which initially proved highly successful at wiping out those resistant staph infections.
However, that victory was remarkably short-lived. As the old adage warns, out of small acorns, giant oak trees grow. In 1961, a highly obscure scientific paper published in Japan reported something completely chilling. The bacteria had already mutated again.
This specific mutation gave rise to the infamous MRSA, methicillin-resistant Staphylococcus aureus. And what MRSA did was an absolute masterclass in evolution. It didn't just try to break our new antibiotic, it bypassed it completely. The bacteria entirely replaced their own structural target enzyme. So instead of using D-alanine to build their walls, which, remember, our beta-lactam drugs love to mimic, they mutated to utilize D-lactate instead. This meant the bacteria simply ignored the beta-lactam drug entirely, rendering a whole class of human medicine absolutely useless against them.
What's really fascinating here is the stark evolutionary contrast between different bugs. Natural selection favors the survival of the fittest, and genetic material can be easily shared between microbes. But while Staphylococcus aureus rapidly evolved, shared these resistance genes like wildfire, and mounted a massive global defense, other bacteria acted entirely differently. Streptococcus pyones, for example, which is another really severe pathogen, has basically never learned the trick. It never developed resistance to penicillin, meaning the original drug works just as well against it today as it did back in 1945.
Part 3. The rise of superbugs. This dynamic, constant back-and-forth escalation of swapping genes and mutating enzymes has brought us directly to the modern crisis we're facing in healthcare today. To put the speed of this evolution into perspective, today over 90% of all Staphylococcus aureus isolates found in hospitals are completely resistant to standard penicillin. 90%! It proves just how terrifyingly effective natural selection truly is when we heavily pressure bacterial populations with antibiotics. And this brings us to the true, grounded definition of a superbug. It is not some new, mythical plague beast with enhanced superpowers. It is simply an everyday, opportunistic bacteria that has acquired a library of resistance to our standard first-line broad-spectrum drugs. The danger isn't that they're inherently deadlier, it's that they force doctors to guess wrong in the initial treatment phase. Lab tests take days. In severe infections like sepsis, missing the mark in those crucial first 48 hours because a superbug just ignored your first-line antibiotic, well, that causes catastrophic delays in effective treatment.
Making matters worse is a pretty harsh reality. The golden age of digging up new fungal weapons from the dirt, that ended in the early 1970s. Despite massive global searches, the pipeline for discovering entirely new classes of antibiotics from nature basically dried up. Today, pharmaceutical companies are largely forced to just play defense. They are endlessly tweaking the chemical side chains of existing drugs, like pushing cephalosporins through 5 different generations, merely to create slight variations that the bacteria haven't seen yet. All of this just to buy us a little more time.
Which leaves us with this final, urgent thought to take away from this explainer. As this millions of years old microbial arms race continues to outpace human chemistry and our heavily modified, borrowed weapons begin to lose their edge, where will we look for the next cure? Will we find a way to use the body's own defense mechanisms? Or are we standing on the precipice of a world where common infections are once again a deadly gamble? It's certainly something to think about.